as Dirk said, I'm going to talk about the neurocognition of developmental language disorder, give a kind of overview on that disorder. So first of all, what is developmental language disorder, or DLD? DLD is usually defined as a developmental disorder of language in the absence of clear explanatory factors such as hearing impairments, environmental deprivation, or gross neurological damage. This kind of definition is not uncommon in developmental disorders. It's very similar in dyslexia. Dyslexia is usually defined as reading impairments in the absence of such clear explanatory factors. Note that DLD was usually previously known as specific language impairment, or SLI. So essentially, DLD is SLI and SLI is DLD, just to make it easier for you to interpret the literature. Okay, there we go. So let's look at the prevalence and impact of DLD. Basically, DLD is quite a consequential disorder. So its estimated prevalence is about 7%. So about 1 in 14 people have DLD, or 2 students in every classroom. It's quite a lot, right? It's much higher than the prevalence of aphasia. It's similar to the prevalence of dyslexia and ADHD, and higher than autism, which is generally thought to be about 1 to 3% in terms of its prevalence. DLD has been found in numerous languages and language families, so it's quite widespread. And it impacts not only children, but also adults, which has evidence for social isolation and lower employment. So again, it's quite a consequential disorder. What does language and cognition look like in DLD? So despite the exclusionary definition, right? Basically, DLD is language problems in the absence of certain clear causes, right? Which might be expected to lead to a phenotypic potpourri, kind of a hodgepodge of symptoms with that kind of definition, exclusionary definition. Despite that, many individuals with DLD show a broadly similar behavioral profile. That is, there's a fair bit of behavioral consistency in the disorder, both in language and in cognition. So particular aspects of language are especially impacted, mainly grammar, across syntax, morphology, and phonology, but also certain other aspects, including lexical recall, so pulling out words. Think of this a bit like word finding. Additionally, particular aspects of cognition are especially impacted. One tends to find this throughout DLD. Executive inhibitory function, so inhibiting, distracting, and conflicting information, working memory, keeping information actively in mind, rapid temporal processing, so particular processing rapidly incoming stimuli, motor skills, particularly more complex motor skills, and procedural memory, which, as we'll see in our research program, we define as the learning and memory that are rooted in the basal ganglia. Okay. There have been 2 main classes of explanatory accounts in the past: linguistic and cognitive, represented here as linguistic and cognitive. Linguistic accounts posit that DLD is due to grammar-specific deficits. So these can account for at least some grammar problems, but not the cognitive deficits that are found in DLD, because grammar, not cognition, is posited to be impaired. The cognitive accounts posit that DLD is due to a processing deficit, generally of working memory, executive function, or rapidly incoming stimuli. These can account for at least some of the cognitive problems, not others necessarily, depending on what's posited to be impaired, but less so the language deficits. However, or I should say, moreover, these accounts focus on processing or knowledge, not on learning, even though learning deficits are quite reasonable, right? Might be expected, at least in part, in a developmental disorder. And this is in contrast with at least adult onset aphasia, where processing or knowledge impairments might be expected, as opposed to learning impairments. Moreover, DLD must be due to a brain dysfunction. It must be rooted in the brain. Like, unless you're an extreme dualist, it's not in the gallbladder or the knee, right? So a neurobiological account should be useful, as with aphasia. So it's in this spirit that we proposed initially in depth in 2005, Ullman and Pierpont 2005, the procedural circuit deficit hypothesis, or PDH. So the PDH is a neuroanatomical account, again, consistent with the spirit of how aphasia is thought of. So according to the PDH, these are the two main points about the PDH. Okay? First of all, DLD can be largely explained by abnormalities of brain structures, again, a neuroanatomical account, underlying procedural memory, in particular the basal ganglia. Remember, procedural memory is defined as the learning and memory that are rooted in the basal ganglia. Okay? And throughout this talk, I'll refer to procedural memory and its neural correlates and associated functions in this kind of reddish color, and declarative memory in this green color, just to make it easier for you. Okay? And the basal ganglia, as most or all of you know, is a set of subcortical, highly interconnected subcortical structures deep in the brain, including these two here, which we're going to focus on today, the caudate nucleus and the putamen. So this is the caudate nucleus. Okay, this is the putamen, and they're loosely linked up front. And together, these are called the neostratum, although the basal ganglia also includes other structures, which I'll mention a little bit, such as the globus pallidus, subthalamic nucleus, and substantia nigra. And number two, the other main claim of the PDH is that the grammatical and other deficits will be at least partly compensated for by declarative memory, which is posited to remain relatively spared in DLD, where declarative memory is also defined in terms in a neuroanatomical way, that is, as learning and memory that are rooted in the medial temporal lobe, including the hippocampus and associated medial temporal lobe structures. So these are the two main legs on which the PDH stands. So let's look a little bit more at the two memory systems, the two circuits, memory circuits, before turning to the more specific predictions of the PDH. So procedural memory, as we've seen, is defined as the learning and memory rooted in the basal ganglia, which again we see here, the caudate, putamen. Together they form the neostratum. Early stages of learning in procedural memory depend on the anterior neostratum. And you'll see this is going to be important. That is the anterior caudate and the anterior putamen. Keep in mind that the caudate and putamen are not don't constitute a fundamental distinction in the neostratum. In fact, in rodents, in primates, you have both, but in rodents, you don't. So it's not that the caudate and putamen fundamentally have different functions, for example. Later stages of learning in procedural memory, as well as automatized processing, depend on different structures. In terms of automatized processing, when procedures or skills are learned in procedural memory, they can be eventually highly automatized. That word is used a lot in this field, meaning they become fast, reliable, and kind of fixed. So at later stages, including automatized processing, procedural memory depends on the posterior neostratum, so posterior caudate and posterior putamen, as well as frontal cortex, including motor and premotor cortex, okay, and parietal cortex. And one way of thinking about procedural memory is that it helps bind neocortical representations, kind of like the hippocampus does, but in this case, primarily between parietal and frontal regions, which then, after systems consolidation, form direct neocortical connections. And those circuits, at least in part, may be thought of as underlying dorsal stream functions. Procedural memory supports the learning and processing of a wide range of motor and cognitive skills. Okay, it doesn't just do some things; it supports a lot of different functions, including sequences, categories, habits, and in language quite a bit. Grammar is best studied, but also other aspects of language. For example, what's usually referred to as word segmentation. So extracting a phonological sequence of word from implicitly from the continuous speech stream, as is often studied in statistical learning paradigms. That seems to depend, at least in part or largely, on procedural memory. So does learning articulatory skills. Also, speech sound representations depends in part, at least, on procedural memory. So it really supports a lot, both in language and outside of language. Declarative memory, in contrast, which, remember, is defined in our research program as an anatomical way, as the learning and memory that's rooted in the medial temporal lobe, okay, underlies and seems to be critical for learning arbitrary associations on items. My favorite example that I try to bring up in all my lectures is that the capital of Burkina Faso is Ouagadougou. Okay? I particularly like that place, so I always bring it up. So if you didn't know that, if you know it now after I said it, you must have learned it in declarative memory. However, declarative memory is extremely flexible. It can really learn pretty much anything, including these things. So it can also support the sequences, categories, grammar that procedural memory supports, although usually in different ways than procedural memory does it. All right, so from this and from the PDH, the basic principles of PDH we laid out a couple slides ago, we can look at the predictions of the PDH, which are that individuals with DLD should tend to show, first of all, anomalies, abnormalities in the basal ganglia and or other procedural memory structures, although perhaps especially in the neostriatum, that's the caudate and putamen, right? Because it forms the sort of root of the of procedural memory. And it turns out that the neostriatum is particularly vulnerable to insult. Anomalies in these structures, in particular in the neostriatum, may be due to a wide range of etiologies, of genetic and environmental etiologies. The idea is, I don't know if you can see me here, is that this is a neuroanatomical hypothesis. Lots of different causes can lead to it. Lots of different etiologies of genetic or environmental insults can lead to these abnormalities in the basal ganglia, okay, which then will lead to a variety of different linguistic and cognitive impairments. That is, impairments of pretty much any function that depends on the abnormal structures, in particular the neostriatum. So in that vein, we expect deficits in language and cognitive functions that rely on the basal ganglia. Pretty straightforward logic, right? Including procedural memory, so learning grammar, sequences, categories, and so on, within language and outside of language, but also other functions that depend on the basal ganglia, right? Because this is anatomical hypothesis. So whatever depends on the affected structures might be impacted. Working memory, executive inhibitory function, and so on. In contrast, declarative memory is posited to be relatively spared and, given its capabilities, may compensate for grammar and other deficits because it can support a lot of these functions that procedural memory also supports. So now let's look at evidence. First, in one slide, is look at evidence for language, then procedural memory, then other cognitive functions, and then declarative memory and how it compensates for the various impairments in procedural memory. And then we'll look at anatomical evidence. So, as we've seen, grammar, in that earlier slide, grammar is impaired in DLD across syntax, morphology, and phonology. Grammar learning is also impaired based on artificial grammar studies, for example. And it appears that early stages of grammar learning are impaired, which suggests, given what we know about grammar depending on procedural memory and the anterior neostriatum being important for early stages of learning in procedural memory, this suggests that early stages of grammar are impaired, that the anterior neostriatum might be dysfunctional. Lexical recall, as we've seen, is impaired, and that, in fact, relies in part on the basal ganglia. And word learning, even, for example, and other functions show impairments when they rely on functions that rely on the basal ganglia. So, for example, word learning is impaired when it occurs in grammatical contexts. In contrast, we find that lexical semantics is relatively spared in DLD, consistent with the hypothesized relative sparing of declarative memory. So let's turn to procedural memory. So when we proposed the PDH first time in depth in 2005, there were no procedural memory studies in DLD. So we predicted procedural memory, that is procedural learning impairments, truly a priori. Okay, there was no evidence for that at that point. Since then, there have been many, many studies, mainly, but not only, in sequence learning, and those sequence learning studies mainly use the serial reaction time task, or SRT task. Okay, and so there have been some meta-analyses of these studies, of these serial reaction time task studies, which have found that, in fact, sequence learning as tested in the serial reaction time task is impaired in DLD. Okay, so here we see the first of these meta-analyses, Ullman et al., 2014. And here's each of the individual studies that were included in the meta-analysis. And here is the weighted average of the studies showing an impairment in SLI, that is DLD. Okay? So individuals SLI perform worse, and here's controls. And other meta-analyses have since then have shown the same pattern. And evidence suggests that there are deficits in other procedural memory tasks as well. So not just sequence learning and the SRT task, including graphomotor learning. This is work mainly by SD Ade Jaffa in Israel, rotary pursuit, category learning, and word segmentation, right? So that's remember, implicit extraction of words from speech stream. And in all these studies, it's early stages of learning that seem to be impaired, largely because they only test early stages of learning, right? You're not going to go on forever, okay? But they are impaired, which again suggests anterior neostriatal dysfunction, because it's the anterior neostratum, if you remember, that underlies early stages of procedural learning. Let's turn to other cognitive functions. So we've actually seen this already. So we've seen that motor skills are impaired in DLD, working memory, executive inhibition, and temporal processing, all of which involve circuitry, at least in part, right? Not only underlying procedural memory, including the basal ganglia. So now let's turn to declarative memory. So declarative memory appears to be relatively spared in DLD, as we would expect, as the PDH posits. So this is clearest for nonverbal declarative memory, but even verbal declarative memory, which of course involves language, which might be problematic, right, given that it's DLD, seems to be relatively spared when one controls for language as well as working memory impairments. Moreover, a fair bit of evidence at this point suggests that declarative memory compensates for the grammatical deficits in DLD. So, for example, individuals with DLD learn and use explicit rules like, oh, if it's yesterday, add ed. And keep in mind that I didn't mention this before, but many of you may know it, that explicit knowledge is learned in declarative memory. One can also individuals with DLD also memorize chunks like walked or the cat as actual words, as chunks, as it's called, rather than putting them together in procedural memory, which seems to take place, the chunking in declarative memory. And as we'll see in the next two slides, there's also relatively convincing correlation electrophysiological evidence for declarative memory compensation of grammatical deficits in DLD. So first, let's look at correlational evidence. So this is a paper by Jared Lum and colleagues in 2012. So what we found was that typically developing children show this pattern that has been found in other studies as well, in typical individuals, that vocabulary, so measure of lexical abilities, correlates with independent measures of learning and declarative memory, tying lexical abilities to declarative memory. Okay? Whereas grammar is tied to procedural memory. This was a serial reaction time task. Okay? So in typically developing children, lexicon is tied to declarative memory and grammar to procedural memory. In DLD, look at this. Not only is vocabulary tied to declarative memory, so is grammar. suggesting that grammar depends on declarative memory, is associated with declarative memory, consistent with compensation by declarative memory for grammar in the context of grammatical deficits in DLD. And essentially, the event-related potential electrophysiological evidence shows the same thing. So in typically developing children, lexical semantic anomalies, such as I like to eat trees, okay? Hopefully all of you got an N400, okay? And you see that here in this study by Fontenot and Van der Wille. Okay? And the N400 has been nicely tied, reasonably confidently tied to declarative memory circuits. In contrast, syntactic anomalies such as yesterday I walk over there leads to left anterior negativities, we see here in blue, okay, which are not tied to declarative memory and are tied to frontal regions and might be, it's less clear, linked to procedural memory. In DLD, both types of anomalies lead to N400s, which again have been linked to declarative memory. So not only I like to eat trees, right, lexical semantic anomalies, but also the syntactic anomalies, suggesting that syntax is being compensated for by declarative memory in DLD. All right. So now we've looked at behavior, okay, and seen that that the predictions are more or less borne out, both for grammar and for cognitive impairments, or in the case of functions that depend on declarative memory, the relative sparing of those functions. Now let's turn to neuroanatomy. So previous empirical studies of the neuroanatomy of DLD have examined a whole bunch of different structures and found at least some abnormalities, some studies in pretty much all of them. Most studies have focused on language and auditory regions, in particular Broca's region and superior temporal cortex, often focusing on laterality differences in these structures, and more recently also examining the cerebellum and the basal ganglia. Qualitative reviews of these empirical studies have argued that abnormalities in DLD may, in fact, cluster in certain regions. That is, there may be some consistency in the anatomical abnormalities, just like there's some consistency in the behavioral abnormalities that we saw earlier, right, in the language and cognitive impairments. And these qualitative reviews have suggested that these abnormalities may cluster, particularly in superior temporal cortex and Broca's region, and possibly the basal ganglia, perhaps especially the caudate nucleus. However, capturing true anatomical patterns is really difficult in qualitative reviews, okay? Since included studies that are reviewed in the qualitative review, differ in various respects that are difficult to capture for qualitative reviews, such as the number of subjects, right? Subjects in more studies should have more weight, as should studies with greater statistical specificity, right? As opposed to those that may be overly sensitive and identify abnormalities in numerous structures that are perhaps spurious, right? False positives. Additionally, many studies examine only certain structures, and perhaps even worse, some structures are examined by many studies. So, for example, lots of studies have focused on superior temporal cortex. So imagine that if many studies have looked for abnormalities in superior temporal cortex, even if a relatively small percentage finds them, if that percentage isn't taken into account, one might get the impression that superior temporal cortex is implicated in DLD. So neuroanatomical meta-analyses can address these issues, these concerns. Okay? And the most common, as probably many of you know, neuroanatomical meta-analytic technique is coordinate-based ones, like activation likelihood estimation, which is the best known. But that's not possible. It's not possible to run AL, activation likelihood estimation, on DLD because there are very few whole brain imaging studies reporting coordinates, and that's required for these techniques with this approach. So we invented a new neuroanatomical meta-analytic approach, which we call co-localization likelihood estimation, or CLE. So basically, the logic is very simple of CLE. We simply compute the weighted proportion of studies finding anomalies in each structure. So say, take frontal lobe and Broca's region, basal ganglia, and neostriatum, for example, right? For each structure, we simply count the number of studies, add up the number of studies that have examined that structure for abnormalities, and then take the proportion that have actually found abnormalities weighted by study sample sizes. It's a very straightforward approach. And this addresses this concern, the number of subjects, okay, because weighted by study sample sizes, as well as the examination of only certain structures, and that some structures are examined more often because each structure is examined independently. The weighted proportion is calculated separately for each structure. It's also very powerful because you can include studies with any method. You can include any study that examines any structure, one structure, the whole brain, doesn't matter. For any abnormality can be included, right? So you can include not only neuroimaging studies and not only for whole brain, but post-mortem studies, anything. So therefore, the number of studies included is large and varied, yielding a higher degree of reliability and generalizability of the results, or likely at least. And then we compute permutation-based likelihoods that the results were due to chance, that any clustering was due to chance, which addresses issues of specificity and sensitivity. So we did this, before I go to the next slide, separately for structural studies, using any technique again, and functional imaging studies. And we published this actually recently, a few weeks ago. So Ullman, Clark, Pullman, Lovelit, Pierpont, Jang, and Turkeltab in Nature Human Behavior, if you're interested in the paper. So for structural studies, we found highly consistent abnormalities in the basal ganglia, specifically in the anterior neostriatum. Okay. Oh, I just saw that I misspelled that. So what did we do? So for structural studies, we examined data in 22 papers, which had 577 participants. Okay, so a very large sample size, of which 250 had DLD and 327 were typically developing controls. And we applied our technique at two levels of anatomical specificity. First, at what we call the first level of parcellation, so 9 structures, the 4 lobes, the cingulate, the insula, cerebellum, thalamus, and basal ganglia. The basal ganglia was the only one to show a very high level of consistent abnormalities. Again, number of percent of studies weighted by sample size at 100%. 100% of the studies showed abnormalities. Nothing came close, only frontal cortex was relatively high at about 80%. At the second level of anatomical specificity, where we basically subparcellated each of these structures, you can see that here, right? Only the neostriatum, okay, so the caudate putamen showed highly consistent abnormalities. And then at a third level of parcellation, only the anterior neostriatum, that is across the caudate and putamen. Note, by the way, when we looked at the caudate and the putamen separately, we didn't show it. Okay? So it's really, I think it's a lesson for any of you who are interested in the basal ganglia. Like, it's not the caudate and putamen may be distinct looking structures, but they're not necessarily the most separated and most functionally in most valid way. So again, highly consistent abnormalities. 100% of studies weighted by study sample found only in the basal ganglia, okay, specifically in the anterior neostriatum. And we performed multiple sensitivity analyses, which showed that the results were robust. So, for example, more stringent as well as less stringent inclusion criteria, also including unpublished studies, dissertations, and abstracts, and also separately examining studies prior to 2005, when the Ullman and Pierpont 2005 PDH paper was published, to avoid bias towards the basal ganglia from that study, as well as after 2005, both of which show the same pattern. This very robust result. Next, we looked at functional imaging abnormalities, okay, using the same technique, using CLE. Again, we found the basal ganglia was implicated. So here we expect less consistency due in the results in the abnormalities due to task dependence of activation, right? If a task doesn't involve the basal ganglia, it's unlikely to show or less likely to show abnormalities in that structure, for example. So this was, we performed the analysis over 11 papers and 414 participants, including 176 with DLD and 238 controls typically developing. And here we found only the basal ganglia and parietal cortex showing abnormalities, although at a much lower rate, as expected, right? Reasons of task dependence, than the structural abnormalities, about 80% for both. And again, multiple sensitivity analyses showed the results are robust. So how can these anatomical results be interpreted? So the basal ganglia and neostriatal abnormalities were predicted by the PDH, as we saw, as were, at least to some extent, more weakly, the parietal and frontal abnormalities, as well as the dearth of medial temporal lobe abnormalities, which I didn't focus on, okay? But there were very few medial temporal lobe abnormalities. The clustering of abnormalities in the anterior neostratum is consistent with what we saw behaviorally, right? Where we saw both early stage grammar learning problems and early stage dysfunction of early stage learning in procedural memory, like with serial reaction time task and category learning and word segmentation and so on. The findings are more broadly consistent with the set of linguistic and cognitive deficits in DLD, all of which have been linked to the neostratum, including, at least in most cases, the anterior neostratum. So grammar, as well as grammar learning, early stage grammar learning, word recall, executive inhibition, working memory, and so on. This is a neat one. At least I find this interesting. So the findings are consistent with the range of genetic and environmental etiologies linked to DLD symptoms, which have also been linked to the basal ganglia, including the neostratum. So, for example, polymorphisms, mutations, right, of various genes, including ANK1, DRD2, CATNAP2, FOXP2, and SEMA6D, have been linked both to DLD symptoms and to the basal ganglia, including the neostratum, as has been thiamine deficiency and prenatal nicotine and cocaine exposure. So again, this is the idea that I pointed out like this earlier in the talk, right? That you have a whole bunch of etiologies, genetic and environmental, which can lead to basal ganglia abnormalities, okay? Apparently, specifically anterior neostratum to a large extent, which can then lead to various behavioral impairments, both in language and in cognition more generally. And so this anatomical count, again, I stress that this is an anatomical account, seems to have greater explanatory power than other accounts, including linguistic and cognitive accounts which can't even explain the other to a fair extent, and of course neither predicts the striatal abnormalities, okay, as well as genetic accounts, because individual genetic counts have less power than this more unified approach which combines them. Let's summarize the neurocognition of DLD. Okay, in this simple summary, we've seen that there's surprising consistency in the language and cognitive impairments found in DLD, which then is consistent with the consistent anatomical abnormalities, which are specifically the highly consistent abnormalities localized to the basal ganglia, particularly in the anterior neostratum. And the PDH, that is a neuroanatomical count consistent with how aphasia is often thought of, has substantial explanatory power for both the behavioral and the neural patterns, more so than linguistic, cognitive, or genetic accounts. So there are a bunch of implications, which at least I find interesting. Hopefully some of these will be interesting to some of you as well. So The results suggest that basic research on DLD should in fact focus on the basal ganglia in various ways. So, for example, might it be possible that DLD in part has a dopaminergic basis, which we know is the case for some other basal ganglia disorders like Parkinson's disease, right? So dopaminergic projections from part of the basal ganglia, substantia nigra, pars compacta in the midbrain up to the neostratum, are critical for basal ganglia functioning, okay? And those are somewhat vulnerable neurons as well. And in fact, there are hints that this could be the case, at least in part. So a number of the genes and environmental insults associated with DLD symptoms in the basal ganglia I mentioned in the previous slide are associated with dopamine, such as FOXP2, catnap2, thiamine deficiency, and prenatal cocaine and nicotine. The direct-indirect pathway should be investigated. I don't know how many of you know much about the basal ganglia, but these are the two main pathways within the basal ganglia that are in fact affected differentially in basal ganglia disorders like Huntington's and Parkinson's disease. Okay. Might these be somehow implicated in DLD? Another question is, there are many genes, of course, that we're aware of, and many of these have been linked to basal ganglia function, but have not been implicated in DLD. Well, those are good candidates to explore to see if they might be involved in DLD. Okay, this is a kind of cool one. So might it be that this way of thinking about it could help explain the higher male prevalence of DLD? So we argued for this in Ullman and Pullman 2015. Basically, the idea is that, so it turns out that females have better declarative memory than males. Okay, I don't know if some of you see where this is going, right? So if girls have better declarative memory than boys, and declarative memory can compensate for many of the deficits in DLD, then girls should compensate more successfully than boys. And we argued for that in this paper. Okay, so in this view, it's not that DLD is actually more prevalent in males. It's rather that girls compensate their way out of diagnosis more than boys. Okay, and we think something similar might be going on for other often comorbid developmental disorders, like we'll see in the next slide, like dyslexia, which also has a higher prevalence in males. There are translational implications of this research for DLD. So, for example, diagnostic, like clustering of anomalies in certain structures and the lack thereof in certain structures serve as early biomarkers indicating an increased likelihood of DLD, for example, in at-risk children. Okay? And perhaps combined with other diagnostic procedures, this could lead to early therapy. And in fact, the research also has therapeutic implications, right? So therapies that target the basal ganglia may be effective, such as dopaminergic agonists, or to encourage compensation in declarative memory, such as explicit input or pharmacological agents that target the medial temporal lobe hippocampal function, such as memantine or cholinesterase inhibitors, for example. The methodological implications. So CLE may be really broadly useful as a neuroanatomical meta-analytic approach, given that it can include a much larger number of studies of different sorts, and thus the results may be more reliable and generalizable than coordinate-based whole brain approaches like ALE. And finally, there are implications for language research. So the findings emphasize the importance of the basal ganglia in language, in particular in language acquisition, particularly in early stages of language acquisition for those aspects of language that depend on procedural memories, such as grammar or word segmentation or articulation, for example. And more generally, the findings support the declarative procedural, or DP model, upon which the PDH is based. The PDH is actually built upon the premises of this model, which we've been working on for decades, which basically simply posits that declarative and procedural memory have been co-opted for language, reused for language in multiple ways, in multiple contexts. And so the PDH is, in a sense, one expression of this. And then last slide, we're also looking at the PDH beyond DLD, although less so. Most of our work's on DLD, including in dyslexia. So that's developmental reading disorder, developmental math disability, developmental coordination disorder, which is basically developmental motor problems, ADHD, obsessive compulsive disorder, stuttering, and Tourette syndrome. And in all of these, the basal ganglia is, to one extent or another, often to a large degree, affected, as is procedural memory. Although in some cases, like in Tourette's, it actually looks like several studies now suggest that procedural memory may actually be enhanced in Tourette syndrome, probably because of, we think, the nature of the basal ganglia abnormalities. So the PDH may help explain a range of the behavioral and brain patterns in these disorders and their comorbidities with one another. A lot of these are comorbid, like, for example, this SLI, DLD with dyslexia, for example, or ADHD, since the same circuitry is affected, right? That makes sense. Although it's becoming clear, at least it seems to me, that the explanatory power of the PDH varies a lot across the disorders. It's not, PDH has less explanatory power for some of these than for DLD, for example, I think. So that's it. I'd like to thank my collaborators at Georgetown and elsewhere, and the funders. This is the collaborators in the broader research program, not just for DLD. And let's always end with a slide. Thank you.
Thank you very much, Michael. I hope you can hear our applause or could see that.
Thank you.
That was a wonderful talk, really clear. What I want to do is start with questions from our own room, and then we have some questions online as well, but let's do that first. So here we go. Suzanne.
Hi, that was a great talk. I'm Suzanne Adloff. I'm in our Communication Sciences and Disorders Department, and I study DLD and dyslexia. So I'm really interested, actually, I'm interested in how they co-occur and how they are actually different disorders. So there are, I'm wondering if you could explain, you mentioned that the PDH may explain how they are similar, but they have different parts of language that are more or less affected. And as far as I know, not too many studies of DLD actually separate out the ones with and without dyslexia. So in part, that could actually affect the results of your meta-analysis. But can you talk a little bit about how you would explain the differences between the disorders?
Great questions. It's more than one question there. So with respect to the meta-analysis, we specifically and very carefully excluded studies in which a substantial portion of the DLD participants had co-occurring disorders like ADHD or dyslexia. So to make sure that the results actually were about, or were most likely about, DLD. So we're reasonably confident about that. With respect to comorbidities, we thought a fair bit about this. We've talked about a little bit in this paper as well as the other papers. So the two main overview papers of DLD for the PDH are the 2005 paper, if you're interested in the Ullman et al. 2020 paper that was in some of the slides in Annual Review of Psychology, if you're interested. Okay, we talked about that in both of those. And basically, it might come to primarily two things. One is that, so within the basal ganglia, there are different, they're called circuits or channels or loops, people use different terms, which are parallel and each of which is connected to different neocortical regions. For example, in the output, to different frontal regions via the thalamus. And we think that one reason you might get different disorders and comorbidities with the same type of abnormalities, so in the striatum, is because different loops are affected, and then comorbidities because nearby loops are affected, right? It's unlike a single that only the loops for, what underlies language and not motor, for example, will be affected. So that's one thing that we think is going on. And then additionally, there are other structures, right? It's not like dyslexia. So even though these results for DLD were quite striking in the specificity of the abnormalities to anterior neostriatum, that's clearly not the case for dyslexia, right? Dyslexia actually, based on other meta-analyses, looks like other structures are also implicated. And in fact, the basal ganglia less so. And so there's a question of other structures also being affected, which can lead to the other symptoms, for example. Does that make sense? Did it answer?
Yes, thank you.
Questions from our room? We have some questions online. Let me start with a question from Yuan. What was considered anatomic abnormality in this study? Difference between, so they're talking about your meta-analysis as well, I think.
Yeah, that's a good question. Yeah. So we purposefully included in our primary analyses any abnormality whatsoever. So whether there was some kind of, abnormal gyri, or for structural hypertrophy or hypotrophy, anything. Okay. And so the analyses I showed you were based on that. That was because although the PDH has hypothesizes where the abnormality should occur, it has not so far hypothesized what type of abnormalities. And so the way I think about this, the analogy I often use is, I can have a swollen hand, I can have a shriveled hand, I can have a bullet hole in the hand, you can smash it with a hammer, I can have a tumor in it. All of those may affect my grasping slightly differently, but they may all affect my grasping, right? And so the nature of the abnormality might not be as important as the fact there's an abnormality. Kind of in aphasia, right? You could have a tumor, you can have an aneurysm, you can have all kinds of damage resulting in aphasia as long as that same structure or set of structures are affected. Now, it looks as though when we look more carefully at the data that in fact that's correct. So no matter what that nature of that abnormality, we have a paragraph on this in the paper, you actually get these similar effects. So it doesn't look like, so I think a lot of us have a tendency to think, just because how we've thought in the past, that, hypertrophy has got to be different than hypotrophy. Well, it may not be the case, and it doesn't seem to be the case in an obvious sense in DLD. We're not saying that the symptoms might not differ somewhat. They probably will, right? But at a gross level, they don't appear to.
We have some more questions online. I do want to just ask as a follow-up to that question and your response. So with your example of the hand that can be injured in many different ways, the functionality of the hand will be impaired in the same way, but the treatment will be very, very different, right? Whether you have a tumor in your hand or whether you knocked it with a hammer. So how does that affect your...
Yeah, it's nice. Yeah. And so, yeah, and I think of that like when I was talking about the dopaminergic possible therapies, right? I said dopaminergic agonists, right? That would only, so if you have these multiple etiologies, multiple causes of the abnormality, right? Then treatments that target dopamine won't affect those that don't involve dopamine. So you're absolutely right in principle.
Thank you. Kirane Tsapkini asks, she says, Michael, thank you for such a convincing talk. I was wondering whether you have looked at the structural connectivity abnormalities of the anterior caudate and putamen as neural substrates of procedural memory. Where do these project in the cortex? Could it be also that frontal lobes are relatively much bigger than anterior caudate and putamen to be compared with? Have you looked at smaller regions within the frontal lobes, such as inferior frontal or middle frontal or superior frontal gyri?
Right. Hi, Kirana. Thanks for your question. So I'm not sure if the question was focused on, if your question was focused on DLD specifically, but I'll interpret it that way. So yeah, so we, for example, if you look, I can actually show you. Yeah, there we go. So when we did subparcellations of frontal, right? So superior motor region, sorry, I think we call this frontopolar, inferior frontal, and then orbitofrontal here. So when we separated these out, we actually did not find, despite the fact that quite a large number of studies examined these, right? So the N was quite large in terms of studies, particular abnormalities in any one of these, as you can see here. I'm not sure that answered your question. I wasn't sure.
We will see what she says in the chat. You have a question from Taylor Barrier. Thank you for the talk. I'm wondering about intellectual disabilities with language difficulties. Would you suspect that there are procedural deficits amongst others in this population?
think I understand the question. So is the question perhaps that for people with DLD who have intellectual disabilities, so lower IQ, for example, um, is that I think might be is that also caused by basal ganglia abnormalities or procedural memory problems? Do you think that was the question?
I'm having some trouble parsing as well, Michael.
So let's assume that's the question, which is a reasonable question. One thing I guess keep in mind that in the new definition of DLD, which was led by Dorothy Bishop, last 8 years or something, it does people with intellectual disability are not excluded, which used to be the case generally with SLI, specific language impairment. So in terms of the origin, which I think was the main part of the question of those intellectual disabilities, I guess it's could be a combination of it's kind of like A and B and then 1 and 2 under A. So could be both the basal ganglia, but also other structures, right? So in the basal ganglia could be both procedural memory and other structures such as other functions such as working memory, executive function, which might contribute to, tests performance on intellectual ability, IQ as well. So I think could be those, but it's not that there are no other impairments elsewhere, right? There are impairments, just that they're highly consistent here. So, and presumably whatever the origin of these, which is quite varied as we saw, probably quite varied as we saw, right, in terms of different polymorphism, different possible environmental origins, those are going to likely affect other structures too. It's unlikely that even if this is particularly vulnerable, the striatum, that's the only structure affected. So I could imagine, I would guess that there are other cognitive problems, including those that are interpreted as intellectual disability, not only from the basal ganglia, but from other structures as well.
Yeah, thank you. Kirana says you answered her question, so that's great.
Okay. I'm glad, Kirana.
Tamar Truzman says, thank you very much, Michael. A plasticity question, please. Is there evidence for anatomical changes in basal ganglia or other regions? And if so, what kind of changes following intervention in DLD?
I don't think I know that. So specifically following intervention, do I know that?
No. Has an effect, has a physiological effect on basal.
Yeah. The best I can do to answer that I can think of right now, at least, I can pull out of my hippocampus right now, is about the hippocampus, which is that there's some evidence that certain therapies, and I remember there's work on dyslexia like this by Guinevere Eden and colleagues, which showed changes in the hippocampus from therapies. And in general, hippocampus can actually get larger with practice. There's some studies showing that. And so that does answer your question indirectly, because I talked about therapies targeting compensation by declarative memory as well, but doesn't answer your question directly because I don't know the answer.
Thanks. I had a question that I put in the chat, which also brings us a little bit to the more regular topic of our series, which is really on aphasia and adult acquired disorders. I do have another question which I'll slip in there because that's a yes/no question. Whether if people with a history of DLD have a higher chance of developing Parkinsonism later on. And then I have an actual question, which allows you to speculate, I hope. If procedural memory problems can at least partly be compensated for through declarative memory, do you think the reverse might also be possible? So we are dealing with cortical damage typically in our groups, stroke from disease. After such cortical damage, do you think procedural memory functions supported by basal ganglia might be able to compensate for impaired declarative memory functions?
Oh, there's some interesting questions here. So the first question, if I understood and remember it correctly, I don't know, and that's a really cool question. I don't know. I don't remember right now at least thinking about this. So I think the question was something along the lines of, do people with DLD have higher risk of, you said Parkinson's, right?
Yeah.
So I guess the logic would be something like, was that you, Dirk, or someone else who asked the question?
Sorry, no, it was me who asked the question.
Okay, so I guess your way of your thinking along those lines is something like, well, if my hand's already damaged, then I'll have more problems if it's damaged again.
Yeah, in a way, yes.
Yes, basically something like that, right? Great question. I don't know. I have no idea. Absolutely none. I you would have to, I guess, retrospectively, you could check that, right, by asking people if they had language impairments growing up, but they wouldn't test for that probably, particularly given how old people are now and what was tested when they were young, but they could give a hint. So I actually had no idea, and I haven't seen anything like that. It's a wonderful question. Um, in terms of the other question, so it's like I said, I think there are a couple of answers possibly. So first of all, in general, I think of declarative memory as a more powerful compensatory system than procedural memory because it's so powerful, because it's so flexible. Like declarative memory can do anything, implicitly or explicitly, by the way. It's not just explicit knowledge. We know that by now. It is known. Procedural memory, I mean, you can absolutely logically turn it around, right? Because procedural memory can do the things that declarative memory can do. Sorry. The things that declarative memory can do that procedural memory does, obviously procedural memory can do. However, it's not the case that procedural memory can do everything declarative memory does, right? Declarative memory is more flexible than procedural memory. So for example, if you look here, declarative memory isn't very good at this. Okay. Yes, both can do this, right? But declarative memory can't do this. So, one-shot learning of the capital of Burkina Faso is Ouagadougou, or, my dog smelled a dead squirrel yesterday in the park. Procedural memory can't learn that as quickly as the hippocampus can, right? So there's limited ability of procedural memory to compensate or to take over at least some of these core abilities of declarative memory. And the other thing just gets a bit trickier, depending on what you meant, is, although both of these are circuits that encompass neocortical as well as deeper structures, right? So allocortical, hippocampus in the case of declarative memory, or basal ganglia in case of procedural memory, they also involve cortical regions. And so if those cortical regions that are being bound by procedural memory, as I mentioned, right, when I talked about it here, right, if those are damaged, then procedural memory may not be able to bind those regions which are damaged. I don't know if that made sense. Yeah. Okay. I think they're really interesting. These are really interesting issues, though, and they're subtle.
We're almost at the hour, but I do want to, if you're okay with it, Michael, I have one more.
Yeah. Fine.
Martina Penke says, thanks, Michael, for your super interesting talk. Could you explore a little bit on the issue, whether in which way the human basal ganglia differ from those of other primates? It's really
great question. Was there more? Sorry. Was that the end?
Yes.
Okay. Thanks, Martina. Hi, Martina. And I assume you're getting at, like, well, if monkeys, I mean, to put it a bit sillily, which you didn't say, but, if monkeys were the same as monkeys in the basal ganglia, why don't they have language? That's one way of thinking about it. I've thought a lot about this, well, a little bit about this. A simple hand-waving answer as well. We have additional stuff, right? We have maybe neocortical stuff that the procedural memory can use in its learning that is critical for language. Another possibility is that there are, which is kind of related actually, that there are loops, there are circuits, channels within the basal ganglia associated with particular neocortical regions, probably particularly frontal regions, that have evolved that subserve language that aren't there in animals, okay, in monkeys, for example. I think that's actually not unlikely. And this is how evolution works, right? You have changes and duplication and things like that. So I think something like that might be an answer. There's also work from Anne Graybiel, which shows that there've been changes, for example, to FOXP2, which lead to more efficient and perhaps different procedural learning in humans than in rodents, which were compared in her work. And so there may be changes between certain species, and I don't know what those look like in monkeys. So it might be that procedural memory, for example, has more capabilities more generally, even independent of the channel that's any additional channels that evolved.
Thank you so much. Anything else from our room now that we have Dr. Ullman? I think we're out of questions, Michael. You've answered everything. This was a really great end to our C-Star season. So thank you so much for being willing to talk to us. Thank you to the audience, and we hope to see everyone in August for season 9 of our series. We keep trucking. So have a good summer.
Thank you. Thanks everybody for attending, and thank you for inviting me. Appreciate it.